Welcome to Ophthalmology Perspectives, the podcast series where we explore the science, strategy, and stories shaping the future of ophthalmology research and clinical trials. I'm Amber Berg, Therapeutic Area Delivery Head for Neuroscience and Ophthalmology at Fortrea, and today we're going to be exploring the evolving landscape of Phase 1 ophthalmology trials with a specific focus on gene therapy and inherited retinal diseases. Joining me are two Fortrea experts who bring deep experience from the front lines of clinical development. David Holscher, a Senior Medical Director, Ophthalmology, who has over 8 years of experience as a Phase 1 Principal Investigator, and Sarah Bulger-Ford, Senior Director of Strategic Delivery and Growth. In this episode, we'll unpack what sets Phase 1 ophthalmology trials apart from other therapeutic areas, especially when it comes to gene therapy. We'll explore why these cutting-edge trials aren't conducted with healthy volunteers and how the route of administration shapes every aspect of study design. We'll share real-world examples and practical strategies for reducing site and patient burden and improving outcomes. Finally, we'll tackle the critical need to balance safety and efficacy in first-in-human trials and look ahead to the innovations like optogenetics that could transform cell and gene therapy in ophthalmology over the next 5 years. So let's get started. I'm going to pose my first question to you, David. What sets Phase 1 ophthalmology trials apart from those in other therapeutic areas, especially when it comes to gene therapy? And why aren't these cell and gene therapy trials conducted with healthy volunteers?
Well, thanks. That's an excellent question. So one of the things that's important to realize is that we want to try to utilize healthy volunteers in development cycles where it makes the most sense and where it's ethical and scientifically relevant to do so, because the costs are so much less. And the ability for a sponsor to get their program up and running with a first-in-human study that's in healthy volunteers, it expedites things and it makes things so much simpler, and it saves a lot of money and time is money in drug development. The interesting thing is that, however, in ophthalmology, things are different. Now, we do have certain therapeutic areas in ophthalmology that can utilize normal healthy volunteers for the first-in-human. Everything else, and most notably gene therapy, we cannot ethically or reasonably utilize healthy volunteers. And the reason is really coming down to also the route of administration. If there's an injection into the eye with intravitreal needles, such as we use in anti-VEGF, diabetic eye disease studies, and then we can't do that in healthy volunteers, regardless of what the investigational product is. And similarly, in gene therapy, we actually have multiple different modes of administration. We have the most invasive, which requires an actual surgery, which places a bleb of the material underneath the retina, but requires a full-on surgical procedure to administer that. We also have the ability to inject into the suprachoroidal space to administer gene therapy, and we also have the ability to do intravitreal injections like we do with anti-VEGF. And none of those routes of administration are appropriate for healthy volunteers. Even if there was such a thing as an eye drop to administer gene therapy, we wouldn't want to give that to healthy volunteers either, because those volunteers wouldn't have the defect in either inherited retinal disease or whatever that need that therapy. So that would never be in healthy volunteers. And then once again, when we get to latter phase, things begin to align a little bit more with other therapeutic areas.
Yeah, David, I'd just add that a lot, one of the reasons for it, gene therapy aside, is to do with the route of administration, isn't it? So, things like glaucoma, dry eye, a lot of that is eye drops, which you can do in healthy volunteers, whereas a lot of ophthalmology indications and treatments require injections into the eye, and particularly gene therapy in ophthalmology requires injection into the eye, and you just can't do that in healthy volunteers.
Yeah, and thank you guys. That was a really great opening response to my question. And I think we can get a little bit deeper into that. David, I'm going back to you, especially based on your experience with these phase 1 units. Can you share examples of how experience and expertise impact the success of these trials, especially given the small patient populations that we're talking about and the high stakes there? Really talking about, especially, with inherited retinal disease, with the younger patient population.
Yeah, that's an excellent point. One of the things that everybody knows in clinical trials, as with other forms of human endeavors, is planning as what you need to do before you can anticipate success. And one of the very important things to plan in these clinical trials is what safety issues that you will tend to have. And this is pretty radically different depending on the patient population that you're talking about. For example, inherited retinal disease patients tend to be younger patients versus chronic diseases like dry AMD and wet AMD that tend to be older populations. So one of the things that's interesting is for the older patient populations, such as with dry AMD and wet AMD, the response of the patients to gene therapy tends to be different than that in the IRD or younger patient population. The older patients don't have as brisk a anti-inflammatory or inflammatory response to the gene therapy as younger patients do, but they do have a degree of intraocular inflammation. What happens to them are other changes in the eye, the most worrisome of which is pigmentary changes. And why are pigmentary changes worrisome as a response to gene therapy is because they can be sight-threatening. And in fact, I had a patient in one of our early trials for subretinally administered gene therapy in a patient with wet AMD who was in her 70s, I believe. She actually completely lost her vision in the study eye due to pigmentary changes that extended into the macula that were a response to the gene therapy. And what we learned from that is that placement of the bleb, which is the little pocket of gene therapy that you place in the retina with the surgery, the placement is critical to mitigate the opportunity for pigmentary changes associated with the bleb to migrate inferiorly into the macula. So you learn to put them on the periphery rather than above the area of central vision, because you can cause, unfortunately, a patient to lose their vision. And for these younger patients, their primary issue with gene therapy is brisk anti-inflammatory responses. And so what that requires is that weeks before you start the gene therapy, no matter what the route of administration is, you start pre-treating them with steroids. So we use subtenon, we use intraocular steroids, and we use oral steroids, and we even use IM steroids because we have to pre-treat these patients before we administer the gene therapy, because their problem is primarily one of intraocular inflammation. And frankly, there's some other problems. These patients have side effects from these high doses of steroids that are given to them for prophylaxis for gene therapy and during and after the administration. And so you can get Cushing syndrome, you can get osteoporosis, you can get blood glucose problems. And so those are all things that you have to anticipate before the trial to plan for the problems that you're going to have. And then with like compliance for ocular administered steroids, the pigmentary changes in the older patients also vary in their patterns and timing depending on the route of administration. The subretinal administration of gene therapy, say for wet AMD is a prime example, those pigmentary changes don't even show up for an entire year after you place the blebs. And this is in contrast to a intravitreal administration of the gene therapy for wet AMD, you see those pigmentary changes almost immediately, and they tend to be more anterior than they do when you put the gene therapy behind the retina. And we're still learning about this, and we're still learning about what is necessary in inherited retinal disease. I'm working on a program in inherited retinal disease, and we are having to use subretinal administration because all of the clinicians and scientists believe that with that inherited retinal disease, that other routes of administration won't work. So this is a work in progress, but we're learning.
It's so fascinating, David, right? All the experiences that we have with gene therapy have given us this insight, especially with the differences in patients. I just find it truly fascinating. Sarah, is there anything that you wanted to add to David's discussion here, or do you want to move on to my next question?
No, I think the only thing that just to go back to the trickiness of the bleb administration and how critical that is, you do quite often find that sponsors are quite hands-on and will sometimes attend those surgeries because, as David said, every patient in a rare disease is critical, and so they don't want to sort of risk it being put in the wrong place. So you do tend to find that sometimes they will attend every single surgery.
The other thing to realize is that these patients are so hard to get a hold of that you need to be flexible in terms of getting them the surgery, because not every site that sees those rare disease patients is capable of the subretinal surgery. And so in those particular cases, what we have is we have sites that have the patients, that refer the patients. Then we have surgical sites where we can transport the patients to the surgical site so that they can get their gene therapy, because just because a site that has the patient can't do the surgery there, we don't want to lose the patient. And this, of course, brings up a whole bunch of logistical complexities. For example, what happens if the patient has to cross national borders to get their surgery? And what happens if the patient doesn't speak the national language of the country in which the surgical site resides? So there's a lot of issues.
Yeah. And particularly in phase one where, you've got so much riding on it.
Well, this is perfect. You actually kind of set me up for the next question that I had for you, Sarah, and going to you, right, from a delivery and strategy perspective. Can you talk about the impact of the trial design and site selection, right? Kind of what we were just talking about, really working closely with these sites, focusing on the complexity and the rarity of inherited retinal diseases.
Yeah, so we quite often say in ophthalmology rare disease that you have to find the patients to find the sites. These are specialized sites. There's not all sites, not all clinics will see these patients. And I say see, not treat, because often there's no treatment available for them. And depending on what the investigational product is, if it is gene therapy, the site has to be able to administer gene therapy, and that requires certification to do that in many countries. And then if going back to the subretinal, it involves vitrectomy. So you need a vitreoretinal surgeon. And quite often, as David said, they're pediatric patients. So then you need a pediatric vitreoretinal surgeon. So, your investigational site pool gets smaller and smaller as you do that. One of the challenges really with inherited retinal disorders in terms of study design is they tend to be slow progressing disorders, and that makes designing a trial challenging. And how do you measure efficacy? If a disease is slowly progressing, how are you going to show an effect? So that in itself can be a challenge. Many gene therapies, for example, require some remaining photoreceptors. So you are looking at younger patients, and then you've got to think, well, can those patients do the assessments? You've got to think about the age. So microperimetry, which is a commonly used retinal assessment, you can't really do that at the age of about 8. So you've got to think about what assessments you are putting into your trial design. Quite often in inherited retinal disorders, we see the use of mobility mazes, which are commonly used as a measure of functional improvement. And you get both physical, and more recently we're seeing virtual reality headsets. Both of these require quite a lot of space and take more than 1 hour to conduct. So again, the site that you select has to be able to accommodate the space, and they have to have enough staff to be able to run those studies.
This conversation is so great because you guys, this is the second time you've kind of set me up for the next question that I had, which was really about the substantial demands within these clinical trials on these, both the participants and the sites, right? I mean, you were talking about space and equipment and, actually spending a lot of time for these participants at the site, maybe going between sites. So one of the most important roles that we play as a CRO is to reduce the burden on all the stakeholders. That includes the participants, site staff, and ultimately, the sponsors. I'd love to hear both of your experiences. I think I'm going to start with you, Sarah, from recent trials where, these burdens have come and how we have worked as Fortrea to find solutions. And maybe starting with some of the psychological and ethical considerations that are really unique to ophthalmology research and how these might impact patient populations, right, on participation.
Yeah, I mean, vision, it's such an emotional thing, so many emotions tied up with losing your sight. It's your independence, your identity, your quality of life. And so patients with eye diseases, they quite often have a lot of anxiety about future blindness, especially if you're sort of getting them early and it's a progressive disease. And they're hyper vigilant when it comes to their symptoms. These patients and their caregivers and their families, because it's inherited, a lot of the time they're inherited retinal disorders, they know all there is to know about these diseases, their own disease, so they're really motivated to join studies out of the hope that they're either going to stabilize and not progress further or have improvements. So they've got such high hopes for treatments, especially like the emerging technologies like gene therapy. But there can be an overestimation of the potential benefits. These are experimental treatments. There is no guarantee that it's going to work. So it's managing that sort of psychological aspects of it that is really important. Patients will trust the relationship with the site, the investigator, the clinician is so important. But equally, there could be too much trust. They say, well, if the doctor says it will be okay, I'll do whatever the doctor tells me to do. It isn't necessarily it is not treatment. It's experimental treatment. There is no guarantee. So, and they've got to understand that. So the consenting process is really important. But there's going to be the fear as well that if something goes wrong, it might ruin the bit of vision that I still have. So that can increase hesitation around interventions. And then with gene therapy, some of them will be afraid of, manipulating their genes and the unknown side effects and what effect that might have on their future children if they are of childbearing age. So there's a whole lot wrapped up in this. And, just to go back to the consenting thing, you have to consider how you're going to consent some of these patients. So yes, if it's vision, that's one aspect, but some of these patients have syndromic conditions that mean they're deaf also. So how do you then consent those patients? There's a lot to think about with ophthalmology that maybe isn't always the case in some other therapeutic areas.
And I would say one of the things, as truly, Sarah, that maybe you want to touch on that I think we do really well at Fortrea is that we really work with patient advocacy groups
Mm-hmm.
to make sure that, we're really taking the patient in mind as we are setting up
Yeah.
these trials, as we're working with our sponsors to make sure that all of those things for these patients are taken into consideration.
Yeah, definitely. Yeah, I mean, you've got to think about the burden, not just on the patient, but also the caregiver, depending on the age of the patient as well. So some of them might be children, they come with their parents, others that need assistance. There might be, as David said, there might be older patients as well. So you've got to think about the burden that it puts upon everybody, the patient, the site, the caregiver. David already touched upon what it effectively is a hub and spoke scenario where you've got the surgery site, but then the sort of more, so that's the central site where they would do the surgery, but then that is not necessarily near where the patient lives, because a lot of them are rare disease, so they travel to these specialist sites, and then that will then put a burden on the patient. So you have what is therefore spokes going out to clinical sites that will do the follow-up, because with gene therapy it's a one-off treatment, but they still have to have assessments for 5 years. These gene therapy studies have a 5-year follow-up for safety reasons. So to make it less burdensome, if they can be followed up at a more local clinical site, that helps reduce the burden. As I say, that's called a hub and spoke, and it gets around the need for multiple vitreoretinal surgeons, for example, as we touched upon earlier. You've also got to think about the time that when you're designing these studies, think about the time involved. There's a lot of assessments in these studies, and they don't just take 1 day, they can take 2 or even 3 days per visit. So that needs to be taken into account. Don't just throw everything at the study design. Do what is necessary, not think about it, is it nice to have? Is it actually going to show me anything? Or is it going to put undue burden upon the patients and the sites? They're tiring. These assessments can be very tiring, which, yes, again, it puts burden on the patient and the site, but it can also impact your data if the data quality can be affected because patients are tired.
Yes, that's correct. Because as we know, there's certain ophthalmic assessments that are very, very dependent on the patient's fixation, like microperimetry. If the patient's tired, they can't even fixate enough to even do the assessment. And the other thing that's important in terms of consenting is these patients need to be aware of, especially in gene therapy, of what they're going to face after those tiring 2 or 3 days, because regulatory requirements require that we follow these patients for a minimum of 5 years. And so that's what is critical for them to understand.
The consenting process is so important because you've got to make the patient aware of the requirements, how intensive these visits are going to be, what provisions you're going to make for their transportation, what help you're going to give them to get to the site, and if they need to stay overnight because it's a multiple day visit, then the accommodation that's going to be sorted for them. You have to help them understand that it's a 5-year follow-up, so it's a long-term commitment. And I forgot what I was going to say.
That's okay. Maybe that gives us the opportunity to move on to the next question. And if you think about it, Sarah,
I've remembered, it was the safety aspect. Yeah. So, and David touched upon it earlier about corticosteroids and steroid treatment. It's understanding what side effects there may be. So prepare them for what is to come in terms of what side effects they might actually experience.
Yeah, absolutely. So, David, I think I'm going to move to you for the next kind of overarching topic here, which is, the risk, benefit, and safety challenges. What strategies do you use to anticipate and manage safety issues before they arise on these trials?
So it's a good question. And once again, it depends on the therapeutic area and the safety population and what safety issues that you anticipate. Inherited retinal disease populations tend to be younger populations. Other gene therapy targets like AMD tend to be older patients, and we know about the safety issues being completely different. And so you need to have pre-op planning. We need to have specific route mitigations, monitoring schedules and contingencies for dealing with breakthrough inflammation in those IRD population and management of the complications for the older patients.
So why is balancing the safety and efficacy especially critical in first-in-human ophthalmology trials, especially those involving subretinal injections?
Because traditionally, also from a regulatory perspective, phase 1 is considered traditionally to be where you're only focused on safety and not efficacy. That's for latter phase trials. But there's a huge stake in manipulating a patient with a gene therapy. And so, once again, no one is going to be willing, especially with a rare patient and an inherited retinal disease, to give this patient a gene therapy and only look at safety. They want to know whether or not the gene therapy is providing benefits to the patient's vision. And so the safety is very important, but also, which is unusual for other phase 1s, we actually have to focus on that because we can't do this without understanding, what the result will be. And obviously, we want to make sure that we balance the need to know whether the gene therapy works as to what problems we may cause the patient from a safety perspective. And once again, balancing that is based on experience and what has been seen in previous trials. So I would say is the most important thing is experience. And so working with partners that have done this before and have seen what happens, because the more you see, the more you see. And the more you do these trials, the more experiences that you have and the better you can plan for the future.
Absolutely. So, Sarah, back to you. Looking ahead, what do you see as the biggest opportunity or innovations that could transform cell and gene therapy trials in ophthalmology over the next 5 years?
Yeah, thanks. So I think for me, one of the most exciting, if not the most exciting thing is optogenetics. So, most gene therapies for inherited retinal disorders at the minute require some remaining photoreceptors to be present, viable photoreceptors. But optogenetics transforms other cells, so like ganglion cells, into light-sensitive cells, essentially photoreceptors. So, and also a lot of the gene therapy that we've got at the minute is usually gene-specific in that it targets a specific defective gene to either correct or replace it. But optogenetics, it's what they call gene agnostic. So it doesn't require an original gene mutation. It works by introducing an entirely new gene, and so it bypasses the need for a specific gene mutation, which means that it can help more patients, not just those that actually have a specific gene mutation. So for me, that is the sort of most exciting thing coming.
It really is, because unfortunately, there are so many different gene mutations, and the traditional gene therapy only addresses a specific mutation. And optogenetics at least has the potential to be more broadly applicable to a patient population.
Yeah, I think it's highlighted, I think, the narrowness of having a gene mutation and the small patient population by the fact we've only got one approved gene therapy for ophthalmology currently, so far, and that requires RP65 mutation. So optogenetics would completely nullify that need. Yeah.
Great. Thanks so much, Sarah and David. That nicely brings us to the end of today's episode of Ophthalmology Perspectives. We've heard how phase 1 ophthalmology trials demand a blend of scientific rigor, operational expertise, and patient-centric thinking, especially as gene therapy opens new frontiers for treating inherited retinal diseases. Our guests have highlighted the importance of anticipating safety challenges, designing trials that truly fit the needs of rare patient populations, and finding innovative ways to reduce burden for both sites and participants. Looking ahead, the promise of optogenetics and other gene-agnostic approaches offers hope for broader, more inclusive therapies, potentially transforming the way we approach vision restoration and disease management. Thank you to David Holscher and Sarah Bolger-Ford for sharing your insights and experiences, and to our listeners for joining us on this journey through the complexities and opportunities of ophthalmology clinical research. If you enjoyed this episode, be sure to subscribe and share ophthalmology perspectives with your colleagues. For more information or to connect with our team, visit the Fortrea website. Until next time.